What's up, guys? How y'all doing, man? We're gonna run through chapter. Yes, I got a haircut. Thank you very much. Let's look at chapter 3 real quick. Not sure if we need to run through all of it all the time, but we're gonna give it a shot and see what y'all think about this. Uh, let's see. It's supposed to be recording now. So we're working here. All right. So chapter 3. Take a look. All right. Now, so chapter 3 is about organic molecules, okay? And it's one of my favorite things. So it should be a really hard one here. If you remember, organic molecules, we haven't, maybe if you read over the chapter, and we haven't covered in lecture yet, we'll talk about this week, are molecules that are made out of carbon. Carbon is the key to life on Earth because it can form 4 bonds on its outside shell. It has 4 electrons in its outside shell. So it can form 4 bonds. So we like to say things that are made out of carbon and hydrogen should be organic. Everything else is said to be inorganic. And so if you major in biology, most of you are taking general chemistry this year. Sometimes that's called inorganic chemistry. And then next year you'll take a year of organic chemistry, 1 and 2, and it is the chemistry of organic compounds, which are mainly those found and made in human or in living organisms, not necessarily humans. Okay, and so I think in culture we say organic is things made without herbicides and pesticides, but in reality it is things made out of carbon and hydrogen. So living things are mainly made out of organic molecules, and all the rocks and gases and water and what, like the ocean is water, which is inorganic. Very important compound for us. We have lots of water in us, but it is an inorganic compound. Okay, all right. And then there are 4 major large groups of organic compounds that we're going to talk about in this chapter, and they are carbohydrates, lipids, proteins, and nucleic acids. Okay, and this is very, very important that you know about these. Okay, so we're going to go into each of these in detail and look at them. Now, one of the more interesting things, if you've ever been dieting, or used to be the old food groups, right? You had sugars, fats, and proteins. Well, guess what? There they are. So we eat and are made out of sugars, and we like to eat fats, which is lipids, for energy, and then we like to eat proteins, which is also a organic compound. So of those food groups, things you remember from school, so we are made out of these. That's why we eat other animals, because they're high in sugars, proteins, and fats. And of course, plants are made out of sugar usually. The 4th one are nucleic acids. We don't really take it for energy, but we use it for information storage, and we'll talk about it. And it also has in its group one of the compounds that carries energy for us, ATPs, or molecules. So we'll come back to it. Okay, kind of introduction, fun one to start off with is this trans fat. Okay, and this is pretty wild here. Trans fats are a type of fat or lipid that do not naturally occur in nature. So it is man-made in the laboratory by adding hydrogen to unsaturated fats, a process called hydrogenation. And it is put in food to increase shelf life and to enhance flavor and texture. But it is not natural. And now we have started doing studies on it. It is probably more dangerous than saturated fats, which are lard and pig butt lard that we fry chicken in, and Crisco and all the things we like to cook in that we know is bad. If it's solid, it's bad. Well, these are even worse than that. And restaurants in California, New York, and California are banning them. And so the FDA is now requiring trans fat labeling because it's such a bad idea. So we made something even worse than saturated fat, which is what's one of the main killers in the South. Right? We like everything fried. Okay, so we already mentioned that what organic molecules are, and then here are the big 4. Okay, let's dive into them. Now, inorganic things are things that don't have carbon and hydrogen in them in reoccurring pattern. Organic things are going to have a carbon chain, right, with these functional groups on them. Now, the functional groups are molecules that most of them contain carbon that are going to be stuck on this carbon chain. Okay, and you can see them here, and I'm not going to go over all of them, but y'all can look over these and even tell you where they're found in and the significance of them, what they do for that compound. And you may recognize some, like amino group is a nitrogen with 2 hydrogens stuck on it. Okay, so these are going to be stuck on the backbone skeleton of carbon, and that's how we make the different organic molecules. Okay, and so carbon, because it can form 4 bonds, is one of the key, is the building block of life on Earth. I would hope that we find life in our solar system before I die, and we get to see what type of molecules they are. If they are based on carbon, then that would be maybe more than a coincidence. Maybe they're kin to us. If they or something else, then that would be really very interesting as a scientist. I would love to see those critters or those microorganisms or whatever they are. Okay? And we talked about these functional groups, and they're going to stick on this carbon chain, right? And we'll come back to that. I mean, it's what it is. Okay, isomers are molecules that have identical molecular formula, but a different arrangement of the atoms, therefore function differently. So here is glyceraldehyde and dihydroxyacetone, and they all have the same number of carbohydrates and oxygens. So they're isomers, right, of sugar and stuff. So, you know, so there's a lot of ways you can stick this together, right? And that's, to me, that's pretty cool. Okay, couple more terms, right? A monomer is a single unit of repeating unit, and if you have a whole bunch of monomers stuck together, you have a polymer. So, for example, sugars are made out of monomers of little single ring sugars like glucose and fructose, and then hundreds of those stuck together form the polymer starch or glycogen. So, just some verbiage here. Matter of fact, amino acids are the monomers for proteins. They stick together in a chain. Like railroad cars on a train, and they form the protein, the polymer, when they all come together. Okay. Big discussion question on our text test on the midterm, probably 10-point question. Describe the 4 organic compounds. List their building blocks or monomers, subunits, and their function. Okay, and what they are, what we use them for in our body. Okay. Now, to build these and break them down, we have two types of reactions that are kind of cool. And these are really cool. So one of these is going to be used to break down the polymers into monomers. And the other one is used to make polymers from monomers or build things up, tear things down. Now think about this. I'm going to home night. I'm going to eat a steak, or I'll eat some food. If I don't eat steak tonight, can't afford that every night. And I will break down those amino acids, take those amino acids into my bloodstream, and then I will use those to build my proteins up, to build my body. Okay, if those that I don't use for energy. Okay, now look at the 2 reactions. They're called dehydration and hydrolysis. Now, if you know anything about dehydration, that is when you pull water out of something, right? So dehydration reactions are going to form water in the reaction, and in doing so, look what's going to happen. You're going to pull water and build polymers. So here are 2 monomers up here, water taken out, and this dehydration reaction causes these 2 molecules, these 2 over here, 2 different monomers, 2 over here become 4 stuck together. So that builds things up. And then when we want to break something down, we add water to it, and we do this hydrolysis reaction. So here we are breaking that one we just built down back into these 2 monomers. So dehydration reactions are going to build things up. Hydrolysis reactions are going to break them down to monomers we can use in our body. And she goes on about this. Others, sometimes we will use an enzyme to help do these reactions. And so enzymes are molecules. We're going to talk a lot about them in chapter 6. But enzymes are usually proteins, can be made out of RNA, that speed up chemical reactions, okay, by lowering the amount of energy needed for that reaction to occur. Okay. Now look at this, almost some kind of sci-fi next couple statements here. But in these assisting these reactions, they speed up the rate at which they occur, and in doing so, they are not consumed in the reaction. They don't give anything up to the reaction, and look at that. And enzymes are not changing reactions, and they don't do anything happen that wouldn't happen naturally. They just speed it up, make it happen faster. And of course, eventually they wear out, and that's why I have these wrinkles up here on my forehead that y'all don't have, because some of my enzymes for repair are starting to break down, because they are proteins and they will eventually wear out. And that's one of the things that happens in aging. Okay. Now, carbohydrates, we're going to look at them then, are a great energy source for our cells, right? Provide the building material for plants. They have a ratio of 1 to 2 to 1 carbon, hydrogen, and oxygen. And they include the monosaccharides, which are simple sugars, single ring sugars, disaccharides, 2 ring sugars stuck together, and then polysaccharides, 10, 20, 30, 40, hundreds of them stuck together. Okay, so a monosaccharide could be a single ring. It could be a 6-carbon ring, a hexose, or a 5-carbon atom pentose. Here is glucose, C6H12O6. Notice all the carbon and hydrogen stuck on it. The hydrogen and OHs, hydroxyl groups, and that everybody's happy with that electron configuration from an earlier question. Disaccharides are 2 of these stuck together, include lactose, which is a glucose and a galactose, sucrose, which is a glucose and fructose, that's table sugar, we put on our cornflakes, maltose and malt liquor and malt balls, etc., is 2 glucose stuck together. Okay. Many people are lactose intolerant, right? They lack the enzyme to break that down, and they have real bad gas and other issues, right? Here we go. There's maltose, dehydration reaction, right? And they're just showing these, gets really excited about those, okay? And then polysaccharides include starch, which is energy storage in plant, and that is what we eat in our bread and biscuits, et cetera, et cetera. Glycogen is stored energy in the liver of animals. And cellulose is the cell walls in plants for their structural material that we don't have enzymes to break that down. So cellulose is something we can't break down that we eat in our diet, and it is 0 calories. It's actually negative calories. You lose energy eating it, and it actually plays a role in helping us with our roughage in our digestive system. But in all diets, eat all the lettuce you want because you can't live off of it. You lose in that. Chitin is in fungi cell wall and the exoskeleton of the shrimp and the crawfish. Peptidoglycan is in the cell wall bacteria. These are all real famous polysaccharides. Okay, look at the structures of these. Here is the starch, that's baked potato, right? Here is glycogen found in the liver, stored in animals, energy storage, sugar storage in the liver in animals. Look at the structural differences. Here is cellulose, and look at this weird 3 strings bound together, and we don't have enzymes to break that down, right? So we can't eat that for energy, okay? Lipids are long-term energy storage, cell membranes, some of the structural components, heat retention. Hormones, a lot of hormones are made out of lipids, and they are for cell communication and regulation of the cells in the body. And of course, some of these are going to be for protection, like waxes. So these include fats, oils, phospholipids, steroids, and waxes. Okay, fats include butter and lard, long-term energy storage. All oils are cooking oil at room temperature, have long-term energy storage. Phospholipids used for cell membranes, also food additive in our food we like to eat. Steroids are 4 ring structures, a group of fats that include cholesterol, which is the main cause of death in America, it clogging up the coronary arteries, #1 cause of death in America. And all the sex hormones are in the steroid group. But not all steroids are sex hormones. Keep that in mind. Of course, drug medicines, and then waxes, including earwax and candles and stuff for us. But earwax is something that we produce to protect the ear and the cuticle of the plant wall, right? And even the surface of fruit have wax on it to repel the water when it rains. Okay. Now, fats are usually made out of a glycerol, a 3-carbon chain, the very simple sugar called glycerol, and 3 long carbon chains called fatty acids. Okay. Now, fatty acids can be all saturated with hydrogen, and these are the saturated fats that have no double bonds in them. And these are lard or solids at room temperature, margarine and butter. And then unsaturated fats have double bonds in these carbons, and that causes the fatty acids to kink, and they are liquids at room temperature. Just like, remember when water packs real tight, it becomes ice and everything. Then water above that, right, it pulls out, it expands, so therefore water floats, ice expands. The heaviest water in the lake, remember, 4 degrees, it's packed as tight as it can, it's at the bottom of the lake. Well, same concept here, these kinky bonds in the saturated, in the unsaturated fats cause this compound to be a liquid at room temperature. And that's pretty fascinating. Okay. And of course, we can do a dehydration reaction and build the triglyceride there on your right. And we can do a hydrolysis reaction, add 3 waters back and break it down into glycerol and 3 fatty acids. It is worth noting that pound per pound, if you had a pound of sugar, a pound of protein, and a pound of fat, the fat has twice the energy in it. So if you're on desert island and there were a pound of meat, pound of fat, and a pound of sugars, pound of sugar, take the fat. You'll live twice long. You'll have twice the energy. You'll live longer than everybody else and outlive them. It just won't be as tasty. I don't really like eating fat, but a big old stick of butter. Okay. Anyway, they also can be used to form phospholipids, which are the cell membrane, main component of the cell membrane. Look at this. So a phospholipid is a glycerol, 2 fatty acids, and a phosphate group. So we draw it as a circle with that charged phosphate group, water-loving polar, and fatty acids hate water. Y'all know oil and water don't mix. And this creates a phospholipid that has a water-hating fatty acid tails and a water-loving phosphate-containing head. So when you drop these in water, they form a bilayer, and this is the main component of the cell membrane. Two fatty acids layers back to back with the fatty acids grabbing each other because they're hydrophobic, hate water, and the water loving, forming the actual boundary with the inside and outside of the cell. Pretty fascinating. We'll cover a whole chapter on cell membranes, little preview of things to come. Okay. Steroids are these four ring structures, right? And actually, it's interesting that cholesterol is mainly found only in animal plants. An animal cell membrane controls the viscosity of the cell membrane stuck in that phospholipid bilayer. It's not present in plant matter. So if you're a vegetarian, you don't get cholesterol, and therefore you don't clog your arteries from the cholesterol you eat. You may clog it from cholesterol that your liver makes. Cholesterol can be converted by the male body into testosterone to cause the beautiful coloration in the beautiful animals, this bird or the peacock, whatever, the beautiful flower, beautiful colors are made by testosterone, which is cholesterol with an oxygen and an OH, an hydroxyl group stuck here. Move that to an hydroxyl group here and move some double bonds around to single bonds here, double bonds and move to here, and a OH group stuck here, and you have estrogen. Very similar, right, structurally. And so the difference in the body is estrogen makes women, and the peacock, not in humans, drab and kind of bland, but also true in guppies, because, if you ever buy guppies, beautiful colored male is $5, $6, $7 a piece, and girls are $3 for $5 at the aquarium store, okay? And now why did God make it like that? Because the male has to attract the female to breed with him, to have to marry him, to have his babies, right? And so the male is showy and has all these colors, and that's why the deer and horns and fight and stuff. Women have been causing fights and all that kind of animal kingdom, not just the humans. I'm just joking on women. Of course, y'all don't cause fight, but that testosterone probably does. Okay. All right. Kind of interesting. We'll talk more about that later. But once again, not all cholesterol, steroids have anything. Cholesterol has nothing to do with steroids, although is the precursor for steroids. Okay. Waxes, long carbon chains here. Look at the wax on those grapes and the honeycomb of this bee. Proteins are made out of monomers. Monomers are the single ones are called amino acids. And there are 20 different amino acids, and the sequence of amino acids in a chain determines the protein. We use these for enzymes in our body, all the metabolic reactions in our body, structural material, collagen in our skin, there's structural material in the body, in the cell, cellular transport. These proteins allow things to interleave the cell. We'll talk about that. Antibodies defend the body, our proteins, and hormones regulate the body, and a lot of them are proteins, are protein derivatives. And of course, muscle is allowed for our muscles are made out of proteins called actin and myosin that contract and allow for movement. So motion is also one of the things we use proteins for. All amino acids, remember, which are the building blocks, monomers of proteins, are made out of these 20 amino acids, and all amino acids have a carbon, a amine group over here, NH2, a carboxylic group, COH, a hydrogen, and then an R group. And it is the R here that represents the variable group that define the 20 different amino acids. Take a look. The circled shaded area is the R group. See the same, look, everywhere you see carbon, amine group over here, carboxylic group, but that R group is different. So here is 5, 11, 12, 13, 14, 16 of the 20 amino acids. I might need to just throw the rest of them in here. I don't know. But anyway, there they are. And you can see these are going to stick together and form a covalent bond called a peptide bond. And the sequence of the order of the amino acids determines the proteins. Now, I use this terrible metaphor, but it's best I can do. R-O-B-I-N, R-O-B-I-S-O-N is Robin Robinson's name. Change it to Z, Zalvin Robinson. It's just spelled wrong. And my name would be Zalvin, not Robin. Proteins, the actual sequence, make the protein. So if Robin Robinson made the Robin Robinson protein, put a Z in it would make it all messed up, right? It would not be Robin, it'd be Zalvin. So it would actually cause that protein to change. So the difference between egg white and Como Steakhouse ribeye steak is the sequence of amino acids in the protein. So it is the sequence that determines the function of that protein, of that actual protein. And it gets a little deeper. You can see that we can do a dehydration reaction and stick them together to form the peptide bond, and hydrolysis break them apart. But it is this sequence, it's called primary level of protein structure. The secondary level is the presence of what are called alpha helixes or beta sheets, where the protein itself folds over. And then tertiary level is an overall three-dimensional shaping of the protein, where different pieces of that one protein will grab each other. And actually look at the picture of it. The sequence is primary. This beta sheeting and alpha helixing is the secondary structure. It may fold over itself. And then how it folds amongst itself is tertiary. But then quaternary is two or more different proteins folded to grab each other, which is pretty cool. So these 4 different levels of protein structure. And notice a lot of hydrogen bonding in there. Holds it. Here's showing some examples of the fibrous proteins and how the perm here versus, and then the spider web shows how the bonding occurs in those. Okay. Another one, kind of an interesting side note they put in here was these chaperone proteins are proteins that help fold the protein so they have that correct shape. And misfolding by these has been attributed to cystic fibrosis, causing a chlorine channel, protein channel that allows things to interleave the cell to be folded improperly, and these people can't get chlorine in and out, and that causes cystic fibrosis. And we've also seen these amyloid plaque buildup in the brain, and some of these others may be caused by a chaperone protein that's misfunctioned that are being linked down to Alzheimer's disease. Another example are pyruns, or misfolded proteins that have been implicated in a group of fatal brain diseases, okay, including mad cow disease. So pyruns are proteins or pieces and structures found out in nature that get in our body and then cause these misfolding proteins that lead to mad cow disease. Okay, so pyruns are believed to cause other proteins to fold the wrong way also. So that is a disease-causing agent called a pyrun. Okay. All right, and our last group is DNA, made out of nucleotides, and the big two are DNA and RNA. And the difference here is DNA has a sugar deoxyribose and RNA has a sugar ribose. But all nucleotides have a phosphate group, a pentose sugar, either deoxyribose RNA or ribose, and then a nitrogen-containing base. There are 5 bases out there. 4 found in DNA, adenine, guanine, cytosine, and thymine. And in RNA, we get adenine, guanine, and cytosine, but we get uracil in the place of thymine. So there are 5 bases, but RNA has a different sugar, and DNA has deoxyribose, RNA has ribose, and then DNA is double-stranded while RNA is single-stranded. And here is the nitrogen-containing base, the sugar, and the phosphate group. Okay, and you can see there's deoxyribose on the right here, and here's ribose. And here are the 5 bases. 2 of these are double ring structures called purines, adenine and guanine, and the 3 pyrimidines, cytosine found in what? Both RNA and DNA, thymine only found in DNA, and RNA, and uracil found in RNA. And if you remember, in DNA, adenine binds to thymine, so purine binds pyrimidine, and guanine binds to cytosine. And so here we got them again, just kind of keep showing all this. Okay, so DNA, a little bit of table there, different sugar, similar bases, double-stranded, single-stranded, and DNA has a spiraling staircase helix shape, and RNA is single-stranded. Here's RNA. You can see it there with the phosphate sugar backbone, with the adenine, cytosine, uracil, and glutamate there. So A binds to T, C to G, and the DNA. And DNA is a spiraling staircase, and there's guanine, cytosine binding there, and then adenine binding to thymine. Notice that C to G is 3 hydrogen bonds, and adenine to thymine is 2 hydrogen bonds. That's some pretty cool chemistry, and you get this spiraling staircase double helix structure. All right. Then ATP is a nucleotide adenine phosphate with 3 phosphates stuck on it. Take a look. So here's the adenine, adenosine, adenine base there, sugar, right? And then 3 phosphates stuck on it. And so when a ATP gives up a phosphate, it releases energy, and we say sometimes it phosphorylates an enzyme or something, thereby transferring energy and making some work occur. So just for you to think about what I'm talking about, you burn ATPs. Pick this arms with 100 ATPs, 1,000 ATPs, whatever. We use ATP as the energy currency to do things in the cellular level. And every cell on Earth, all living cells on Earth use ATP as their energy source to do this work. Pretty fascinating. All right, guys, just a quick overview. We'll hit a little bit more in lecture. But I just want to throw it out there for y'all to look over this so we can kind of blow through this, because it's going to get a little bit different when we get over to energy, where we talk about these and then how they're actually used to do things and how we build things in the chapters coming up. Y'all have a good evening. I'm going to see y'all tomorrow. See y'all in class. Bye-bye.